Welcome back. This lecture is about the use of location sensors and in general for health, most importantly, public health, meaning health of a group of individuals. We'll start by talking about the radios that these wearable mobile devices have, and these radios are the sensors that we use for location tracking. Some work better outdoor and some will work better indoor. In this particular lecture, we will mainly concentrate on the outdoor localization and in general public health on a global scale. And so we'll talk mainly of GPS, and I'll describe what this is, but most of you already know Wi-Fi and cellular example. In the next lecture, we'll talk about Bluetooth and PAN, also RFID. Technologies are very different in the way they are architected, but also the way in which the signal is transmitted and their energy efficiency levels. And I'll try to give you some hints also of the system perspective every time we touch one of the new technologies. So I believe all of you who have a mobile device is familiar with GPS, which offers localization outdoor. And GPS is the word used, is the acronym of Global Position System, which is generally United States governed. However, there are other satellite systems governed by other countries, and here are a few names of them. And in general, GNSS is the Global Navigation Satellite System, which kind of comprehend a series of these. So in general, we should talk about Global Navigation Satellite System to be country independent. In reality, we all talk about GPS in practice. So in GPS specifically, there are 30 plus satellites. All satellites are very sophisticated and have an atomic clock. Obviously, we'll talk about our receiver. So let's consider when I talk about receiver, imagine your mobile phone. Generally, they don't have the level of precision or the clock as the various satellite. We'll hint at how the temporal aspect can be resolved. The position of the satellite is known very precisely. Again, I have another slide on this. But the time delay between when a satellite transmits a value, a message, and when the receiver receives the message is obviously proportional to the distance between the satellite and the receiver. Slides again on this. And if the satellite broadcasts the message also containing the time of its transmission, then if you go back to your very basic physics, you can understand that you can figure out the time of travel, the time of delay, so the distance between the two, and therefore help your localization, because the travel time per speed of light is equal to the distance. Now, how do we find the travel time? Well, again, if you can read my handwriting and if you can understand what this is, well, I'll tell you, this is my image of a satellite. So the satellite transmits this message, and in the message, it would say the time of transmission is T0. So I transmitted this at my T0. And then here's your receiver, which is really badly drawn again. And so the receiver will know the time of arrival, the time of reception of the message. And so here, time of arrival, which is the time I know, let's assume this is precise, and I can perhaps say a bit more how you can get a precise clock by simply having more information about your various satellites and receiving that information and then using that to recalibrate your clocks, or even you can do it with base stations on the ground. And again, another slide on this. So you have your time of arrival, you have the time of sending the message, which is indicated in the message itself, you have the speed of light, and you can calculate the distance, your distance from the satellite. Now, can this be used to understand your location? Well, another very badly drawn diagram. If the world was flat and you have, let's say you have a satellite here, a satellite here, and a satellite here, and you somehow have a distance, you are at a certain distance from each of these three, well, there's only one point you can be. If you think about this example a bit more. So let's say if you only knew your distance from Rome and Venice, then you can be in these 2 points. Well, let's use that example. If you know the distance from Rome and Florence, then you can be in these 2 points. But then if you also know your distance from Venice, then you can only be here. Now, this is true for a flat world, but in 3D, you also need another sphere. And the sphere is the Earth. This is the fourth sphere. And you can, I don't want to start drawing or gesticulate over 4 spheres while I present this, but you can reassure yourself that at the point with the 4 spheres in 3 dimensions, you can just get 1 point, and that's your location. And that's how the satellites, in very brief and simplified form, find your location. How do we know, though, where the satellites are? Because you need to know where they are to kind of decide where you are. And so satellites are on fixed trajectories, and there is a book of satellites and approximate orbits information that can be downloaded on the receiver. And ground control station compute corrections and satellite trajectory, and so these are updated on the receiver. Now, you might have heard the word assisted GPS. And you might have also noticed that if you don't have assisted GPS, you have a cold start. You have a period where you go out with your device. Maybe you're too young for it, but I had a device that didn't have assisted GPS. You go out with your device, and the device tells you for a while that it can't find your location. And that's because it's trying to download this almanac of locations before it can start being working. Assisted GPS is simply a way of saying that the almanac, that initial startup part of the work, can be downloaded through a different network. And in fact, a Wi-Fi or cellular network can be used to download this initial information needed for the GPS to work. And that's all it is. And the drawing on this little corner just explained that your phone is here in the corner, and it's connected to a base station, which is connected to a backend, which receives this information from the satellite about their positions, and you can start having this information already sent in this way, and then you can really localize yourself by knowing the positions of the satellite and how far you are from them. Now, involuntarily, by talking about satellites and assisted GPS, we started talking about cellular networks. So we've seen GPS is based on satellites. Cellular networks somehow are also used for localization, not just for communication. Cellular networks are the base station, are built by this concept of base stations, and you are receiving information through the base station on your phone, through the cellular radio on your phone that connects to this base station. Every time you move from one base station to another, you have a handoff to a different base station. In the same way that you can localize yourself, you could use that to localize yourself. So if you're receiving from a certain station, you're certainly in an area that corresponds to that station, but you might be also receiving some signal for a different station, even if it's further away, and that can be used to add further localization information from your phone. The density of base station changes depending on population. Obviously, in London, in certain very used parts of London, the density of base station is much higher than here in West Cambridge. And so depending on that, you might be able to say you are at a certain distance from an area, or localize yourself better or worse. In the next 2 slides, I have a few examples of the kind of information you can build over the data of mobile phones and GPS, as well as localization information. So this is movements in Tokyo of people using different mechanisms of transport. And this is, the changes of color correspond to how they use, this is the time, 4 p.m., people are starting traveling again, but shops and services are very used. Some people are eating. And then in the evening, people are traveling back and using the information. Now, how is this built? Well, there are services that build on localization information, things such as Foursquare, and they use the people location and the information that the user sends through the app, for example, saying, I am in this location, and this location is a restaurant. And so by GPS and information of the category of the place, this kind of maps can be built. And these are very important usage of space maps. There's another one here. This is one that one of my ex-postdocs built. This is Manhattan. The circles are essentially, I will animate it soon, but I want to tell you before, the circles are essentially, the radius of the circle indicates how many people are in that particular central point of the circle. The colors indicate various categories. For example, we have entertainment in red, college and education in black, shops in white, food in yellow, parks and outdoor in green, travel in light blue, nightlife in purple, and home in blue. And so if you see, the thing you can notice while the time changes is the difference in colors and even in dimension. 6 a.m., this is travel. Some of the stations are really busy. And some other college and education came up a bit earlier. These are eating places. And then in the evening, lots of purple because people go to eating and clubbing. And then travel becomes a big thing later in the evening. So it's kind of obvious, but it gives an idea of how people use the space and when they use the space. The spatio-temporal dimensions of this are very important. There's a large literature using this type of data to understand behavior and to then use it. And we have a few examples of this, especially the second example on COVID-19 is one that you will find particularly linking to these last two data sets animation. So the first example is about the mobility and dementia. So Alzheimer's disease is a very common disease that affects people, and there's no cure for it. The only thing that can be done early on, if it's spotted early on, is to slow it down. So the earlier, the better. So how do we detect Alzheimer's disease? The current techniques to detect Alzheimer's diseases is very invasive, is lung puncture tests, so it doesn't scale. Researchers have found that a particular portion of the brain, the medial temporal lobe, that also contains the hippocampus, is responsible not just for memory, but also for navigation, and is one of the first that is affected by Alzheimer's. And so I describe one of the studies that has been done. In fact, it's a study that we have participated on, where 33 participants at mild cognitive stage of dementia, so reasonably early on, so we're still going around, were given GPS devices. These are just device trackers. So they were a mobile phone, there were specific devices just containing a GPS tracker. There were many, there were 15 patients and 18 controls, and they were tracked for 15 days. So here are some examples of the tracks of the patient and the controls, in fact. And so one of the things that we know is that trajectory and navigation, returning to particular place, is driven by a part of the medial temporal lobe called entorhinal cortex, which is one of the first things affected by Alzheimer's disease. And so looking at trajectories, looking at how these location segments where the features of this location segment and try to compare the ones of the patients and ones of the healthy individuals is a very interesting problem. So here's an example of a segment that was returning perhaps home or to a residence within 10 meters of it, and it could take 1 to 20 minutes. Over this segment, certain features were calculated. For example, many segments were really similar to each other, so across the kind of same parts of the space. How discovering of the space there were, so entropy, how much distribution of the cells where all the segments of a person covering. How far was someone going from home? How many stops did they make, and how much were the stops? And then looking at the angles of the trajectories, because the complexity of the trajectories indicates perhaps that someone was lost or making something too complex. And so I don't want you to understand all the details of this analysis, but again, you have the metrics here, which you know very well, sensitivity and specificity. We won't talk about uncertainty of the prediction because I've not introduced it. 1 to 3 are at the bottom, segment similarity, you've seen it in the previous slide, segment similarity, duration of stops, and entropy. And it turns out that if you look carefully at this picture in your own time, you will find that these 3 metrics together were very good features in predicting if a user was Alzheimer or a healthy individual. So together they had a combination of sensitivity and specificity. Some other metrics work well for one of the 2 metrics as well, but this was a good combination. I know that this is a very small study, and this is just preliminary to give you an idea of how location data can be used in the context of neurological diseases. A final example is we have all the participants on the x-axis, and the blue are the healthy one and the red are the patient ones. And you can see that the prediction of being healthy, or being a control, is quite high for most of these, getting it right. It gets it wrong for a few of them, considering various tries of the data. And here, for 2 patients, it's clear that we're getting them wrong. We think they're healthy. Now, interestingly, it turns out that these 2 patients are the ones that have the mildest cognitive impairment, meaning that they were almost healthy, they weren't quite healthy. So somehow, while this is an error, it's an error that indicates that perhaps there is a scale. And so the fact that this predictor could perhaps have shades of gray, and obviously with bigger data, maybe this leads to something more interesting. This is a very open area of research. There are interesting questions of how many samples do you need from a patient to understand their behavior? What are the features that are really discriminative? Well, I gave you a list in a table, but there's so much more that can be looked at. And what stages can we discriminate? We only tried to say this is AD and this is healthy, but can we discriminate early and later stage? We saw that we got 2 patients wrong because we were just trying to do a binary classification. Can we do something more refined? Now, we'll talk about the elephant in the room, which is privacy, especially for location data. This is very important later in the slides today, but one of the questions, do we really need to send the user's data elsewhere for analysis? Or can we send just the features, or can we run the model on device? These are all very interesting questions we can discuss perhaps in class in the questions, or perhaps you can look at the later slides and we can open the discussion later. The second work I would like to talk about is one related to COVID. This, I notice now for you that this was published in November 2020. So essentially the first year of COVID. And it was quite novel. This was partly in Nature. It was quite novel for the predictions that they came up with, I think, at the time. Now, some of these things are known. For example, they came up to the fact that restaurants were really an important place where a lot of infections was happening. And so that'd be probably the biggest thing to keep closed for longer. So at the time, I think it was a very important. So here's the typo, March 2020, no, March 2002. So this should be swapped. This is the data. The data was taken from an aggregator that took data from things such as Foursquare and other apps that collect where people are over GPS data, as well as indicating the type of point of interest they are at. CBGs are essentially equivalents of neighborhoods, I would say. And these are really just the demographics, but not demographics, the numbers of data that they have. Now, I would like you to understand how they drew this network. So POI, point of interest, essentially restaurants, gyms, the train station, everything, is here geographically described on this purple graph. So this is really geographical. The description is a map of the point of interest. At the bottom, geographically equivalently, on the green, these are the neighborhood that we have. And all these lines here are indicating the movement. The thicker the line, I'm not sure you can see how thick the lines are, indicate how many people from this neighborhood in reality go to these places. You see the lines are generally vertical because people in a neighborhood tend to stay in the neighborhood and use the resources of the neighborhood. However, there are quite a few mobility patterns that are diagonal, especially in the middle here. You can see a lot of a mixture as well. Now, if I ask you to indicate the differences between this graph and this graph, I'm sure you can immediately highlight the fact that in this graph, there are so many less gray lines, meaning that there was much less mobility, everything was locked down, right? So it's already visible in the network. So the data represents a good representation of what happened on the mobility of people at that time. And now here is probably the most difficult part of the lecture. I'm trying in a few slides to give you an idea of how an epidemic model works and how we can use it. I have a few formulas in the next slides, and I'll try to explain every little bit a bit intuitively. But essentially, a model has a bunch of individuals defined as susceptible, people that could get COVID, exposed, people that have been infected but are not yet infectious, then the infectious one, and the recovered, which can be either recovered or removed in some other models, meaning that they didn't make it. In some of the models, you can go back to susceptible. In this particular model, although it's perhaps not realistic for COVID, the model didn't go back to susceptible. I think at some time scale, this is acceptable because you are recovered and you might not be infected for COVID for 3-4 months. So this is a realistic model to apply in this sense. The model that they built has only 3 parameters: the transmission rate, a point of interest, and I'll explain how they decided which transmission rate they had. One can imagine that perhaps a restaurant should have a higher transmission rate than, I don't know, a train station where people just meet each other very quickly. A neighborhood has a transmission rate, which is probably very similar to each other. It's just you are in the same area. Somehow it might be dependent on that fact. And the initial proportion of individuals that have been exposed to COVID. These are the only 3 aspects of the model. And these are the formulas, and I will describe every single one. I know they look very daunting, but they're actually not very difficult to understand. And so here, this first one indicates the number of individuals that at time t move from being susceptible inside this neighborhood, this CBG, as they call them. So how many people become susceptible in this CBG at time t from susceptible to exposed. So they're healthy and they become exposed to COVID. They're not yet infected, but exposed to COVID. And this is the composition of 2 distributions. One is a Poisson distribution, the other is a binomial distribution. Now we can argue why they've used Poisson and binomial, but I don't want to get into that. But essentially, the Poisson distribution will give a probability of having a number of people exposed from the fraction of susceptible time. This so I go to the next slide because the next slide has the annotations and might help you. Sorry, my annotations are here. They're pretty, I hope you can read them. But this is the susceptible at this neighborhood C at time t. multiply by the rate of infection at this POI, PJ at time t. So you iterate over all the POI, all the places, and consider that every individual which is acceptable in your neighborhood C might have a chance to go to these POIs that are visited and get exposed. So how do we know how many people go to this specific POI? Well, we have a matrix that says how many people from this PBG go to this POI. How do we build that matrix? Well, if we go back to this graph, you have that. Over time, you can aggregate and say, well, roughly, these people from this place go to this with a certain probability. So you can find these probabilities automatically. And so by considering this rate of infection of the POI, and we have a formula here that indicates that, we'll talk about that later. So if you know how likely you are to get infected at that POI, and then you consider the fraction of people that from that neighborhood go to that POI, and that's what you define here. This is the fraction, the visit matrix, and then you multiply by the fraction of people that you have that are susceptible, then you have all of a sudden a distribution of the people that can get exposed when they visit a number of POIs, all of the POIs, because there's a sum. At the same time, there's also a probability of getting infected inside the neighborhood, which is independent on the POI. And that's the rate of infection at the neighborhood, defined as being in the neighborhood. These are very similar. So they are the rate of change of exposed to infection. And really, these are just binomial. They consider the means of the latency period. So how long would it take for this disease to go from infected to infective? Same for recovery. So these are very simple. So I said that this value here is the rate of infection to POI. How do we find that value? And this is here, and this is essentially beta, which is the value of transmission somehow at that POI, which is dependent on how many infected you have at time t over visitors at time t. So if you have the infected and visitors, then you have another probability that depends on the area of the space. There's a transmission context that they have. There's a dwelling time. How much, this is very important, how much are people staying in this place? And that might depend on the type of the POI. The total number of visitors spread over an area. And so this really depends on how big this POI is, not just on their type. So by using these parameters, and you might not have understood all of the types of parameter, but essentially, I would like you to get the full ideas. And this shouldn't be daunting if you ever see an epidemics paper. They essentially consider little variables that are associated to how big a POI is, what type of POI that is, and what's the type of interactions that people have in there, considering how much people of a certain neighborhood visit that type of POI in particular. And that gives you the whole formula for this. How well is this model predicting the data? Well, in general, on the y-axis, you have the confirmed cases, which obviously went down in every city. The reported cases is this orange line. And the model itself, for all the cities and in general here, seems to follow very well the curve of the reported cases. So the model seems to follow the data. This simple model with just these three equations and some parameters seem to follow the data very well, which is a good thing. So things can be studied perhaps in the future using a model like this. If you have data until now, you can look at the future. So this is another part of the study that looks at the infectivity per POIs according to their model. So according to the model, only 10% of the POI give you already an 85% of the fraction of infection. So one might think you don't have to close this full city, you just have to close certain type of things. And proportion of infections in different cities also say that. here, restaurant. Look at Chicago, restaurant. So you get a feeling of what perhaps you want to close. Here is an example of the number of infections. Sorry, the number of infections over the fraction of the visits for partial reopening. So if you close, let's say 10% of this, you would have 50% of infections. So if you open here, you have 40% of cumulative infections, 40 over 100,000. If you instead open only 10%, then you would have a 50% infection, and that's quite low. But you also perhaps could look at what to reopen, because the additional infection compared to not reopening, if you reopen restaurants, this is very high. So if you don't reopen, you have much less. But new car dealers, convenience stores, you might as well keep them open because they don't add much. So this is a very interesting finding. One of the aspects of that was studied in here was the relationship between income and race effects on COVID. And here, looking at predictive mobility, you notice that the bottom income decile, which is information that was contained by neighborhood, like a neighborhood, you know by the census that you know this kind of demographics data, and then you will see that two curves with that some people would move more in with respect to the time. So the purple line is higher, and also on the other graph on the right of this, you see that those places that have the purple are the places where the infectivity, according to the previous graph, was higher. And the people with lower income tend to visit those places more often than the people with the higher income, unfortunately. The other two graphs per city that you see, A and B on top there, essentially talk on the y-axis about relatively infection risk. So in Philadelphia, in general, you find that there is much more risk for lower infectivity, lower income than higher income in getting in really getting infected. And if you're lower income, you get infected more than you are if you are higher income. The third example I want to give you is one that uses cellular data in general for public health related to malaria. Malaria is carried by, well, a mosquito, but it's spread by people. So it's the malaria parasite is carried by an infected person that then get bitten by a mosquito and then bites another individual and transmits that parasite. And so mobility studies are very important to understand how malaria spreads and how it can be confined. Reference at the bottom. So let's look at this. I always like graphs, so let's look at these two graphs. At the bottom, the first figure indicates the base station. So this is cellular data. You have base stations in all these areas. Obviously, not all of this country is covered. And the red areas are the red where malaria is endemic, where there's quite a bit of it. And the gray dots are the settlement of people. Out of this data, there have been some clustering of these gray dots into groups that tend to be in similar areas and with the same kind of settlement properties. And so they've clustered the areas in this. And out of those colored settlements, you see some are in those red endemic areas and some are not. Out of these, if you remember those colors, you can see that the colors are reproduced in these in this two panel. The first graph is what we call a movement graph. So the nodes here are just corresponding to the different clusters. You can ignore the numbers here, they're just different neighborhoods. Some have the same because they're part of a bigger area. But what you see here is the lines that are thicker if you have lots of people. So in this example, you have lots of people going from this yellow to this blue. And that means there were lots of people doing this and going back here. You have thinner lines, but this is essentially the mobility graph as per the cellular data of people across the different neighborhoods. Now we know that some of these neighborhoods were malaria endemic. And so you could study importing parasite from region to region. And so the second graph is a graph where their annotations are related to returning residents. So they were studying people going somewhere and then returning from an area that had endemic malaria. And so where are these returning arches coming from? And again, if you look at the correspondence from graph A and graph B, it obviously changes, you have much less, but you have a lot of returning from this yellow one to this, this blue one. And although if you look in the first graph, there's a lot of mobility this way, well, because this wasn't an endemic kind of area, then the important arrow is this one that shows the flow of the parasite. And so this is very important information because it allows physicians to then decide on the interventions and where the intervention should be in particular. We know where the endemic parasites are, but we don't know where the people are going from those points. Now, in this last part of the lecture, I want to talk about ethics and privacy that touch all of the data that we are seeing in this general course. But because location is very sensitive, I think you have it high in your mind, and I thought of linking it to this lecture in particular. So anonymized aggregations of location data offer some protection. And in fact, one of the papers even that we talked about today says that they exclude census block groups, so neighborhood group, if there are only less than 5 devices recorded from that census block group to a POI. This is to protect people from being anonymized because the anonymity group of 5 is too small. People could be, and I'll talk about how you can do the anonymization on this later. If you want an example of location data de-anonymization, this is an example. So if you have information about a user's home and work data and work location, then it is likely that you might be able to look at this gray curve. So census tract is equivalent to a zip code, and this is a county level. If you have information of the home and location at census tract level, so at zip code level, if you have both of them, for a fraction of the population, you have 40% of the population for which you can say that they are likely to be 1 in 10. So if you have this, you have an anonymous user, you know their location zip code for home and their location zip code for work. This study shows you that actually only 1 in 10 people in that particular study they had could correspond to this. So that's quite worrying. However, it's much better to have information at the county level only, because for, let's say, 30% of the population, you're already completely anonymized. So the set of enemies is much higher. So you're one of quite a lot of people. But why would you be able to de-anonymize? Well, I want to describe a specific study that was conducted by machine learning challenges. So Netflix data. Netflix is a distributor for content, and data from Netflix about anonymized, let's say, likes of movies was a data set of that kind of movie X liked by this anonymous users or unliked by this anonymous users was released to improve machine learning algorithms. But someone has shown that by that data, by using additional data set for IMDb, where perhaps users' information was known, you could de-anonymize some of the users that had more niche kind of preferences. And this created a big problem, in the community. One thing that happens is that the sensing data, so this is just movie preferences. So sensing data, including location data and other data, has a much wider variety of preferences, so types. I go to the gym a certain time during the day, and then I have a train ride at a different time. Perhaps I even do this on different specific days. And so the data sparsity of all this is very high, which means that the ability to localize, to identify someone with additional auxiliary information that I might be able to have, maybe through Twitter to public data feeds, is much higher. And this is a paper that does something similar. In the graph, essentially, it shows that depending on how much data you manage to have, you might be able to identify someone, users, let's say for a K equal 3, a group of a limited set equal 3, with 20% of some sensing streams, you might be able to identify about 60% of the users. And that's quite high. You can read the details on the paper, but essentially saying sensor data, location data, and sensor data, even just accelerometer data, is very sensitive. And so it needs to be treated with care. And so all of the techniques and approaches that might be able to give you the ability to calculate machine learning on device, federated learning, the ability to take away as many features, calculate the features locally, are all very good techniques to use when you're dealing with this sort of data, especially if your health is starting to be considered. This is just for your information, another set of papers that you might be interested in reading in this domain. I won't talk about them. And with this, I will conclude this lecture. And if I find my mouse, I will say bye-bye.